today what I want to do is tell you about what Darwin didn't know. But let's start on a more positive note and take a minute to celebrate Darwin. So as an evolutionary biologist, I always like to start talks with Darwin, but especially, of course, on Darwin Day, as we're all here, as Jeff mentioned, to celebrate his 209th birthday, and of course, the 159th anniversary of the publication of his most famous book, On the Origin of Species. And I think it's fair to say that very few fields, in the way that evolutionary biology can trace its sort of origins to a single man, and a single book, and a single elegant idea, like we can in evolutionary biology. Today what I want to do is talk about Darwin and everything he knew and all his prescience, because it was remarkable how much he knew, yet with so little information, at least relative to what we know now, and how much he got right. Okay? And really, the main contribution that Darwin made through careful observation and detailed experiments was to put forth this idea of evolution by natural selection. The idea being simply that in a population, if you have a number of individuals, and those individuals vary in some trait, and that trait confers some advantage to that individual such that they survive to reproduce and pass on more genes. And if that trait is heritable, then that population is subject to evolution by natural selection. And Darwin spent years documenting variation among individuals, documenting how those trait variations affected survival and reproduction. But there was one piece sort of missing, and that is the heritable component. Now, he recognized that many traits are heritable based on the resemblance between parents and offspring. But what was missing from his grand theory was the mechanism by how traits were inherited. And this is arguably one of the few things that Darwin got wrong, because he was pressed to come up with the mechanism to finish this sort of grand theory. And he proposed an idea which now to all of us seems a little bit ridiculous, because of course at the time this was unknowable. And he showed deep regret in coming up with this mechanism sort of on the fly, because as we all know, Darwin spent years and years documenting things and very careful about what he proposed before he actually eventually submitted his book. So today what I wanted to do was to link Darwin's great discoveries with that second great discovery, that is the three-dimensional structure of DNA. So in 2004, a historian of science, Matt Ridley, made this nice connection. So what you're looking at here, I don't expect you to read, but maybe you could just sit in awe for a moment, because what you're looking at here is Darwin's last publication. It was published just 3 weeks before he died in a little-known journal called Nature. So, of course, Nature is still one of our most prestigious journals. And this paper is entitled On the Dispersal of Freshwater Bivalves. And really what it is a natural history observation of a freshwater beetle, or a flying beetle, and clamped to its leg is a cockle, or a freshwater clam. And this is a report of that natural history observation. So you may be wondering, well, why was this published in Nature even in 1882? And it turns out, this is a little bit of an aside, that this solved a great mystery of how cockles could go from freshwater pond to freshwater pond because they could hitchhike on the legs, for example, of beetles. Okay? So it solved this great mystery. But what's more relevant to today's story is how Darwin got his hands on this natural history specimen. Well, it was sent to him by a young naturalist who was a shoemaker by trade. They had a flurry of correspondence, not by email, but by letters. And this young shoemaker's name was Walter Drawbridge Crick. So to some of you, that may ring a bell, because nearly a century after the publication of The Origin, that young shoemaker's grandson and his friend Jim Watson, with key insights from Rosalind Franklin, made that second great discovery, the three-dimensional structure of DNA, providing a mechanism by which traits could be inherited, and what Jim Watson humbly referred to as the secret of life. And it's in this DNA text where we get even more evidence for Darwin's great theory, the idea of our 3 billion year existence, the shared evolutionary history of all living organisms, and what I want to talk about today, the mechanistic details of how organisms adapt to their environments. So our lab, like many others, like Darwin, is very interested in understanding how diversity in morphology and physiology and behavior evolve. But thanks to Watson and Crick, we can use information from the genetic code to answer that question. So today, the main question that's going to sort of encapsulate what we're going to talk about today is trying to understand what the DNA base pair changes are that are responsible for traits that matter for organisms in their natural habitats, that allow them to survive and reproduce and pass their genes on to the next generation. Now, in recent years, and Jeff alluded to this, there are many groups that have been trying to make this connection between genes and traits. So to illustrate this, I just want to provide a quick example, and an example that I think would have really delighted Darwin. As many of you know, Darwin had a fascination with pigeons. You just have to open the Origin of Species, start reading the first chapter, which is almost completely devoted to pigeons as a subject of domestication or breeding. And he, in fact, was so inspired by pigeons and breeding that he wrote a second book, and these are original images taken from that second book. And so why did he talk about pigeons? He talked about pigeons because he used this as an analogy to natural selection. So he said man can select from an ancestral form. So like this rock dove, let me grab this stick. Yep, giant lightsaber. So here is this ancestral form, and man could select on little variations in that form and produce pigeons that had tremendous feathers or puffy chests or weird bills, and etc. And so the analogy was that now if you just remove man as the selector and let nature do the selecting, that's where you have natural selection. And this type of analogy was really effective in putting forth his theory. So now what we know, based on, again, the foundation of DNA and genetics, is that folks have been able to go back to these pigeon breeds that so inspired Darwin and identify the genes that are responsible for these morphological variants. So for example, we now know the gene that gives this white penguin its dilute color, or genes that give pigeons an even distribution of pigment, or these beautiful wing bars that we see, or the head crest that we see in some pigeons. And my favorite, we know 2 genes involved in foot feathering in pigeons, so why they have feathers on their feet. So this is just an example. There are many other examples in which we now know the precise DNA base pair changes and how they work through development to produce morphological variation. However, if we think about other traits, like behavior, for example, we have many fewer examples. And this is what I want to talk about today, using the same sort of approaches that folks have used to figure out why we have different colored hair, or why some pigeons have feathers on their feet. We can apply those approaches now to ask about what are the genetic changes responsible for behavioral variation. And in fact, Darwin was also quite fascinated by behavior. So now if we go to the Origin and flip to chapter 7, he spends a whole chapter describing hive-making bees and slave-making ants and asking this question, can instincts or behaviors, just like morphological traits that we saw in pigeons, be acquired and modified through natural selection? And this was a question that really troubled Darwin. In fact, he said, if this wasn't true, this would be a difficulty sufficient to overthrow my whole theory. But if you read through the chapter, he concludes in his eloquent Victorian prose that indeed behaviors just like morphology can evolve through natural selection. So we're going to talk about one approach to finding this link between genes and behavior. So why, you may ask, do we have so few examples? We know behaviors are really important for survival and reproduction. And I would offer two explanations, or two hurdles that make this challenging. First is we need to choose behaviors that are highly heritable, have a strong genetic component that gives us power to try to find the genes, and behaviors that one could argue on average are more influenced by the environment, and so it's a harder job than trying, you have less power to find the underlying genetics. The second challenge, I think, is maybe even harder, and that is simply how do we measure behavior? So it's quite easy, one could imagine, to measure how many feathers are on the feet or the color of pigments in feathers. But by contrast, behavior is harder, and I want to illustrate that with this example. So Drosophila are model organisms. Folks have been studying Drosophila, in particular Melanogaster, for many years. And arguably, one of the best studied behaviors that's stereotyped and highly repeatable is courtship behavior. So right away, you'll be able to see I'm not a Drosophila biologist, but let me describe how their courtship goes. So you put a male and a female. The male's there on top. He has a slightly darker abdomen. You put them in a tube or a petri dish, and what will happen is they'll orient to each other, and then the male will move to the back behind the female, and he'll tap at the female, sort of poke at the female. That's step 2. And then after he's done poking her abdomen, he'll go back around to the front of her and start singing to her by vigorously flapping his wing. Then there's some licking involved. We'll skip that step. Then some attempted copulation, all before the deed is done. Okay. So now let's think about how we quantify behavior, not just in an individual, but let's say variation between individuals or among species. So what do we measure? Is it the angle that they orient that's important? Is it how many times he taps her, where he taps her, how hard he taps her, how he sings, how many vibrations of the wing? So already I hope you can get the sense of measuring behavior can be quite challenging. So one of the things that has happened in the last few years is that there have been these new advances in automated behavioral analysis where computers can track individuals and do all the measurements for you in a robust, reliable, and unbiased way. Okay? So this allows us to get over at least that one hurdle. Or I'm going to tell you about another way in which we try to circumvent this challenge of measuring behavior. Okay? And that is, we could take advantage of an idea that was first proposed by Richard Dawkins in his second most famous book. Some of you might know The Selfish Gene. This was a second book published in 1982 called The Extended Phenotype. And in this book, Dawkins put forth the idea that if an animal has an instinctual or innate behavior that results in that animal producing an artifact, and I'll give you some examples in a minute, then we can treat that artifact just like any other phenotypic trait in our body, like the length of our femur. Okay, so let me give you some examples to highlight what we mean by an extended phenotype. So for example, this is a bowerbird from Australia. This particular male of this particular species will collect these sticks shown here, put them in this particular shape. This particular species also tends to decorate its bower with blue stuff that it finds in its environment. Once the bower is built, the male will strut back and forth in this bower as a way to attract a female. So this is a behavior he doesn't learn from his parents, but one that's thought to be highly genetically driven. And within the species, all the males build the same type of bower, and other species, they built different shapes of bowers. So if this behavior of building a bower is controlled by genetics, then we can use the bower as the trait of interest and treat it just like hair color or feathers on pigeons' feet. So here are some other examples. These are nests built by swallows. Different swallows build different shaped nests out of different material and put them in different locations. Spiders, often different species of spiders build sort of stereotype shape of webs, have them hang in specific areas. And then a final example, one that's slightly more complicated because this is an extended phenotype not built by a single individual, but in this case a colony of termites. So today I'm going to tell you about one particular type of extended phenotype, and this is the burrow. Here's just an example of a diversity of species that build burrows, from bivalves to ants to tortoises and prairie dogs, with more or less realistic depictions of those burrows below. So clearly, burrowing has evolved in a number of different species independently, which suggests that burrows have some sort of function. And you can already start to imagine, I'm sure, the types of functions that burrows might serve and how they relate to fitness. They can be important for avoiding predators, for thermal regulation, avoiding extreme temperatures, for those species that meet and mate underground, social interactions, for food, storage, or growth. So clearly, burrowing can have an impact on survival. The other reason we were interested in burrowing is because quantitative geneticists have shown that certain components of burrowing can have a heritable phenotype. So using laboratory mice that they brought into the lab, put in a giant box of dirt, they could look at the burrows that mice built. So here's an entrance tunnel, and this is the excavated area. They showed that certain components of burrowing could be highly heritable. And then my favorite example that is not super relevant to today's talk, but I think is really cool, is that burrows and burrowing behavior can fossilize. So this is a fossil from South Dakota, which is referred to as the devil's corkscrew because of this shape. For many years, nobody knew what produced this kind of burrow, but in fact, it's a burrow that's produced by a now extinct giant beaver. So behaviors can fossilize when they're in the form of an extended phenotype. So we didn't start to study burrowing behavior in extinct beavers, but instead we turned to this species here. So I want to introduce you to today's main player. This is a mouse called a deer mouse. It's in the genus Peromyscus. They're the most abundant mammal in North America. And while to many of you this looks like a standard mouse, in fact, these guys are actually outside of the split between our standard laboratory models of mouse and rat, which are separated by about 10 million years. These guys are about 25 million years from those two groups. So they really represent their own unique lineage. The reason that we study these particular mice is because, like laboratory mice, we can bring them into the lab. They breed really nicely. We can do controlled experiments in controlled environments. And our group, along with others, are generating a lot of new genetic and genomic tools, plus we can borrow a lot of tools from laboratory mice and rats. But here's the kicker, which is how they're different from laboratory mice. These guys are not human commensals, so they're not found primarily, at least, around houses. They're found in natural habitats across much of North America. This is the distribution of just one species. You find in the top of the Rockies, the deserts of Arizona, the coasts of Maine, surely on Long Island. And because they live in so many different habitat types, this certainly contributes to the great amount of genetic and phenotypic diversity that we see within and between species, including behavioral variation. So depending on who you talk to, there are about 55 species of these particular mice. But today I want to start by introducing you to one of them. This is Peromyscus polyanotus. It's called the old field mouse. And it's called the old field mouse because it primarily occupies open old fields. Okay, not a particularly clever name. And here I've highlighted two of our field sites. So here is one of our less glamorous field sites. So this is a burnt peanut field in central Alabama. By contrast, here's one of our favorite field sites. This is the beautiful white sand beaches of the Gulf Coast of Florida. And what these two habitat types have in common is there's very little vegetation. So these mice are running around in areas where they experience high levels of predation. This particular species is not just my favorite species, but was in fact the favorite subject of study by this fellow here, Francis Sumner. Francis Sumner is a classic natural historian who drove around much of the US catching mice and describing variation. He's shown here in all his sartorial splendor. So this is what he wore when he would go out in the field to collect mice. We barely dress this nicely to give lectures. And he published a number of papers, but one paper in particular, a little-known paper that was published in 1929 with his graduate student, was particularly influential. And I have to say, I like to show this picture. This is his grad student here. because all of us know that given enough time, graduate students will converge in their phenotype on their graduate advisor. And I think this picture nicely highlights that phenomenon. Okay, so this particular paper is a natural history note and describes a very interesting behavior performed by this species of mouse. So what they found was if they go out in the field, they would find that this species would build burrows that look something like this. You would find an entrance tunnel, and you could tell it was an active burrow because it had this sort of plume of freshly excavated soil. And if you dug down, there might be a little plug, but this would be a tunnel that went down to a nest chamber. And then they had this secondary tunnel that radiated up towards the surface, but importantly didn't penetrate the surface. So next what I want to do is tell you a little bit about how we catch these mice in the wild, because it's going to give you two key insights to this burrow construction. So what we do is usually around, let's say, February, when it's miserable in Cambridge, Massachusetts, we get on a plane, go down to Florida. It's not hard to find field assistants. We drive around, look for these old open fields because these mice live exclusively in these open fields. We walk around and then try to find burrows like this hole here, and that's that plume of freshly excavated soil that suggests it's an active burrow. The next thing we do is we get a graduate student and we ask them to lie down on their belly and put their face in the entrance of the burrow. Okay. Then we stand around and watch this, laughing gently. So what my graduate student Jesse is doing is he has plastic tubing, and he's taking that tubing and he's weaving it down the entrance tunnel till he hits the nest chamber. Then once he hits the nest chamber, he blows through that tubing, rattles it around, disturbs the mice inside. Then what happens? We have an important job. We're standing around. We're literally jump on the mice that come flying out of that secondary tunnel with the hopes of catching those mice. So I'm telling you this for two reasons, both because I think, one, it's cool, right? And, but two, we can't see that secondary tunnel where that comes out. Remember, it doesn't penetrate the surface. We only know the entrance tunnel. But the size and shape of these burrows are so stereotyped that we can predict with great accuracy where that secondary tunnel is. And the second thing is that secondary tunnel acts as an escape hatch, such that if something comes in the entrance tunnel, like our plastic tubing, or a predator, they have a backdoor escape hatch to get out. Okay? And because we can predict with great accuracy where that tunnel, escape tunnel comes out, that allows us, even people like me, to be able to literally pounce on these mice, which you have to trust me, are very, very fast. Okay? So why might they have such a complicated burrow design or ingenious? So as I mentioned, they live in these open habitats. They're subject to a lot of predation. So having a sophisticated burrow is probably a good investment. But they also, in these open habitats, are subject to predation. One of their primary predators are snakes. Okay? So if a snake comes in that entrance tunnel, it's nice to not be trapped and have a way out the back. But you don't want that backdoor tunnel to be open because then you're subjected to predation from both sides. So the other reason we got excited about these burrows, clearly they could be ecologically relevant, but also because of work done by this fellow here, Wally Dawson, in the 1980s. What he did is he took these mice from the field, brought them into the lab, and showed that they would build burrows. And they would build burrows even if they had never seen dirt before, for sometimes tens of generations, which suggests this isn't a learned behavior, but in fact maybe innate. So what we did is we returned to the same sort of approach that Wally did now something 40 years later, and we built these giant boxes in our animal facility so we can control temperature, environment, food, soil moisture as much as we could. And in each of these boxes, which we refer to as our phenodomes, in these phenodomes, which are quite large, there's about 1.5 tons of dirt in each one. We put in this dirt and we give them a slope because the mice like to dig into the side of the slope, and this prevented them from digging in the corners. And in each trial, what we did was take an individual mouse, put it in one of these boxes, and left it for 2 days completely on its own. And then we would come back after those 2 days, trap the mouse out, and now we would measure, this is where we got really high tech, measure the burrows using what we refer to as phenofoam. So phenofoam is the stuff that if you have a hole in the wall, you buy at Home Depot or Lowe's, you squirt it into the wall, it will fill that space and harden. And so now, using this phenofoam, what we can do is make a cast of the burrow. And what that cast represents now is a physical representation of the behavior that that mouse performed over 2 nights. So this is our extended phenotype. And let me tell you, measuring one of these casts is much easier than measuring the mouse's behavior directly. So next, what I want to do, let's see if this works, is show you how we catch these mice in the lab just like we do in the field. Oh, we might have to skip this. Okay, we're going to skip this. But what you would see is this little, right here is the little burrow. We put the tubing down through here. And what you can see is there's no escape hatch here. And what you would have seen is out of nowhere, this little mouse's head would pop out, right? I know, I'm sorry that you're going to miss that, but trust me, it's very cute, and you can't, the main point is that you can't see the escape hatch here. So then for all the mice that we put in these assays, we measure their burrows, and today I'm going to focus on just some very simple measurements. We're going to talk about the entrance tunnel length, the total length of the burrow, and the presence or absence of this escape hatch. Okay, so the first question we had was Polyanotus, we know from those natural history studies, build these burrows, and we showed that in the lab. But what about all the other species of Peromyscus? So using the same approach, we tested a whole number of Peromyscus representing the diversity within this group and asked what kind of burrows do they make in this standard assay. And the first thing we found is that not all mice burrow, and there's a lot of variation among species. So here what I'm showing you in green is the frequency at which they burrow. That is how often they burrow when they get the opportunity. And in gray, when they do burrow, what is the length of the burrow? So you can see some mice, like Polyanotus, the one we've been talking about, builds often and builds long burrows. Some other species do similar types of burrowing, but some don't burrow at all. And in fact, this was quite nice to see because Peromyscus eremicus is a rock specialist. It doesn't build burrows in the wild, and it doesn't build burrows in the lab. The second thing I want you to note is that there's a very strong correlation between the frequency of burrowing and the total length. Okay? But that's just the length of the burrow. What about the shape of the burrow? So we focused on Polyanotus, these ones that build, these are not to scale, but cartoon depictions, right, of an entrance tunnel or a nest chamber or escape tunnel. But Maniculatus builds just these little stubby sock-shaped burrows. So does Leucopus, that, or little divots. This Aztecus, this one, produces long burrows, builds them often, but it's just a single tunnel that goes down. Sometimes it forks off, but never an escape tunnel. And these guys don't build burrows. All of this gets much more interesting when we look at this in a phylogenetic context, so we consider the relationships among these species. Because what this suggests, for example, is that even closely related species like Maniculatus and Polyanotus build very different burrows, even though these are, in this case, sister species. This also tells us something about the evolution of the traits, like the escape tunnel is unique to Polyanotus. So when we think about the escape tunnel, what we're looking at is a gain in complexity of burrowing shape. So for the rest of the talk, I'm going to focus on these two sister species, because here they're very closely related species, yet produce these very different burrows. So the first thing we wanted to ask about these guys is how much of this burrowing behavior actually looks genetic. As I told you, these mice, we can put them in to one of these boxes. They often immediately start burrowing, having never seen dirt, having never seen another mouse dig a burrow. That suggests there's a strong genetic component, but we wanted to test this more rigorously. So what we can do is take Maniculatus, the little burrowers, and Polyanotus, the big burrowers, take their little jelly bean-shaped pups, and then swap them such that they're raised by the opposite species as parents, and then test them when they grow up at their burrowing and ask, does it matter who raises you, and what does that affect on your burrowing behavior? And the short answer is there was no measurable effect. Maniculatus digs little burrows when it's raised by maniculatus parents, and it still builds little burrows even if it's raised by the big burrowing parents. And the same is true for the other species. Note the axes are different here. So that gave us some suggestion that in fact genetics may be important here. So this then gave us the confidence to start doing genetics and do some crosses. So what we did is we took maniculatus and polynotus, put a male and a female of one from each of the species into a cage. When not given a choice, they happily will mate and produce fertile and viable offspring. So we're generating hybrids. And next what we did, generated those hybrids and asked what kind of burrows do hybrids make? So you can take a guess in your own head. I can tell you in the lab that we were all making bets. Money was exchanging hands. Drum roll. So think about what you would guess, what kind of burrow they would make. And it turns out F1 hybrids make perfectly complex burrows with a long entrance tunnel and a full and complete escape tunnel. You can see that with this data here. So here's entrance tunnel length, little burrows, long burrows by the parents, and the hybrids make burrows like polynotus. Even more striking is the frequency of escape tunnels. Maniculatus, these guys, never make an escape tunnel. Polynotus always does, and F1 hybrids always do. Okay? So what this says to me as a geneticist is that the complex burrows, these types of burrows, the genes or alleles that are responsible for that are dominant. The complex alleles are dominant. Okay? That was really cool and surprising. And this also informed the next step, which was to take these F1 hybrids that build polynotus-like burrows and mate them back to maniculatus over here. So we're going to take polynotus-like diggers and then give them another burst of maniculatus DNA. In this next generation, the genomes get shuffled, and on average, the second generation of mice are going to have 3/4 little digging alleles and 1/4 big digging alleles. And we can look at what types of burrows they make. So here we're going to focus just on the length of the burrows, shown here. Here are our parents. Maniculatus make little burrows, polynotus big burrows. I already told you F1 hybrids, their children, make these polynotus-like burrows. And when we take these guys and back cross them to maniculatus, the next generation build these sort of a whole range of burrows. So if you remember back to Genetics 101, this looks very different than Mendel's peas. It's not a 1-to-1 ratio. It's not a 3-to-1 ratio. Instead, we see this continuous distribution. So what that tells us is that it's not one gene controlling this behavior. Perhaps not a surprise. But what was really interesting to us is that we were still getting mice that built polynotus length burrows over here. So what that means is that it's not 100 genes either. And you can in fact use this distribution to estimate or guesstimate how many genes are involved. And this estimate is about 3 to 5 genes. So to me as a geneticist, that's great. 1 is a little bit boring. 100 is intractable, so somewhere right in the middle. Okay, so this is what we learned about burrow size. What about burrow shape? So excuse my ability to draw in PowerPoint, but you'll get the picture. So when we looked at those bat cross animals, you'll see that some built these little maniculatus burrows, some built polynotus-like burrows, but really interesting, some built long burrows without an escape tunnel, some built short burrows with an escape tunnel. What that means is that you can genetically separate the length of the burrow from whether or not it has an escape tunnel. What that means is that those alleles are not the same gene and in fact are probably on different chromosomes or on different ends of a chromosome. Okay? Then the next thing we did was to ask a very simple question. What fraction of these bat cross animals had burrows that had no escape tunnel versus those that had an escape tunnel? And when we counted up the number of individuals in each of these categories, what we found is there were about 140 mice that had no escape and 130 with an escape. This was a dream result. This is much more like Mendel's peas. Here we have a 1-to-1 ratio. which at least is consistent with the idea that the presence or absence of the escape tunnel is consistent with a single gene of major effect. I should note there are also much more complicated alternative explanations, but this is my favorite. Okay, so now that we know that burrow length is maybe controlled by a few genes and presence or absence of escape tunnel may be as few as one, we wanted to know where are these genes in the genome. So next what we did was took all those back-cross individuals, we know all of their burrow shapes, and now what we're going to do is we're going to genotype them at lots of markers across the genome. So all those mice, back-cross mice, 380 of them, we have their burrow shape, we genotype them at lots of markers, and those genotypes tell us for each individual what parts of the genome they have from Polynotus, the big diggers, and what parts they have from the little diggers. Then we simply ask, are there regions of the genome that are predictive of burrow shape? So if you have a big digging allele at the end of chromosome 1, do you always dig a big burrow? If so, that means there's a gene at the end of chromosome 1 that probably controls burrow shape. So we do this throughout the genome, and here's what we find. So these mice have 23 chromosomes. I'm just showing you 4 of them here. Think of this as the association between genotype and phenotype, and it has to cross this significance threshold. In black, I'm showing you entrance tunnel length, and in red, escape tunnel. So what you can see is that there are 3 chromosomes that have a statistical association with entrance tunnel length. And and that's perfectly consistent with what we predicted. I said 3 to 5 genes or loci associated with length of the tunnel. We found 3. And escape presence or absence, we have this one association on linkage group 5, consistent with potentially one major effect locus. And third, I told you we could separate length from presence or absence of escape tunnel, and that's because the alleles responsible are on different chromosomes. Okay? So we got really excited about these results. But depending on whether you're an optimist or a pessimist, you could say, you know, some of these peaks are barely crossing significant threshold. And we sort of thought about that too, like, is this not a screaming signal? But here's the results that convinced me that we found something that I think was really tractable. So remember now, the Polynotus alleles, the complex burrowing alleles, are dominant. So if we compare mice at each one of these genotypes that shows the strongest association, those that have 2 maniculatus alleles, and compared to one that has a dominant Polynotus allele. When we do that, the average burrow length goes up by 3 centimeters. Similarly, in linkage group 2, if we put a Polynotus allele on an otherwise maniculatus background, the burrows increase in length on average by 3 centimeters. The same thing is true for linkage group 20. This is not Photoshop data. And I have to say, this was completely not predicted. Oh, I can stop screaming. Okay, great. So what this suggests is that each one of these loci acts independently. In general, gives you a 3 centimeter increase in length in an additive fashion. And if you add these 3 up and you have all 3 QTL, you have a Polynotus allele that pretty much makes up the difference between the length of Polynotus and maniculatus. Okay? Similarly, if you take this marker here and you compare the 2 genotypes, you go from a 20% chance of making an escape tunnel to an over 50% chance. Okay. So now we got really excited. There's something there. And so for the last bit of the talk, I'm just going to focus on this linkage group here because it's the biggest and it's the one that repeats the most. What we really want to ask is what are the genes in this QTL peak that are actually affecting entrance tunnel length? Okay? But as you can see, this peak is pretty big. It basically encapsulates half a chromosome. So that's like thousands of genes, and we may be fishing for just one. This is the hardest part of this approach, is how do you go from thousands to 1? So what you can do is you take the best burrowers in that first back-cross generation, back-cross them again to maniculatus, take the best burrowers and back-cross them again, and that narrows in on your region. And then you can take that big peak, and it gets narrowed to something that looks like this, which is much more tractable. When we ask how many genes are in this little region, there are only 54 genes in our QTL peak. And it turns out this is a hotbed of olfactory receptors. While it could be an olfactory receptor that affects burrowing behavior, We didn't think that was quite obvious, so we just put those to the side for a minute. So now we're left with about 25 genes. Of those 25 genes, we asked which of those are expressed in the brain. It turns out these 8 are expressed in the brain. Okay? Then we asked, of those 8 expressed in the brain, which ones are expressed at different levels between big burrowers and small burrowers. And it turned out that just 3 of them were. When we looked at these expression patterns more carefully, this gene, Chrome5 or M5, was a real standout. So let me tell you about why. M5, this particular gene, is different. So here we're looking at expression levels. If we look at Polyanotus, the big digger, and compared it to Maniculatus, there's a difference in expression. If we looked at the expression of the F1 hybrid, what we see is the expression is at a similar level to Polyanotus. Remember, F1 hybrids make burrows that are similar to Polyanotus. The final thing we did is you could look in a hybrid that has both alleles, Maniculatus and Polyanotus, and ask, do those alleles in the same genetic background show a difference? And it's the Polyanotus allele that shows higher expression. So this got us really excited. We had a screaming candidate gene. Okay? Now we thought about, how do we test this gene? Okay? We predict at best it's going to cause a 3 centimeter difference in burrow length. And it turns out that's just a little difference in the way we were assaying behavior. So we decided that, despite the fact that I think I convinced you that measuring mouse behavior directly was hard, that we had to stop beating around the bush and had to look directly at mouse behavior. So how do you do this? This is a mouse behavior that occurs at night, in the dark, and underground. Okay? So luckily I had a very smart postdoc in the lab. And what Brandt did is he designed what we call the Brandt Farm, which is akin to an ant farm that some of you may have had when you were kids. So think about taking a slice of one of these burrow boxes that's about 2 inches wide, made out of clear plexiglass on either side. And in doing so, now what we could do, if we did this under IR, a red light, we could actually watch the mouse doing its behavior. So keep your fingers crossed. See if this works. Okay, it's going to be green for a second. Hopefully it'll come back. So you can see so this was so fun. You could see what the mouse was doing, and even just watching the first few videos, we learned all sorts of cool things. Like the mouse gets down here, and to build its nest chamber, it turns over on its back, starts digging up. You can see it kicking the sand out because it's going to build that big pile of sand that's going to come out. Every once in a while it gets tired. It will come up tired. I don't know if it's actually tired, but every once in a while it'll come back up. It seems to take a little break. It will survey the land around. It'll clean up that loose soil that's in the tunnel and continue. So we got so excited about, like, now we can see the mouse doing its thing. But then we realized that each burrowing assay is about 8 hours long. Each mouse we videotape 3 times, and we have very eager Harvard undergraduates. But that's a lot of hours of video to watch when we're talking about hundreds of mice, okay? And then there's the problem of what do we actually, even if we had all the time to watch these videos, what do we measure? Is it how many times they go in and out, how many paw flicks they use, how many times do they turn on their back? We're back to that problem of how do we measure behavior? Okay? So as I mentioned before, there are these new approaches to automate behavioral analysis, and this is what Brandt did next. So here what you're watching is a mouse digging its burrow over 8 hours, sped up 900 times. So this is in the ant farm. Here's that slope. The mouse has dug its burrow right away, so we're watching this in time. And over time, we're keeping track of 2 things. The amount of soil that piles up out here, which is correlated with the amount of soil that's missing down here. So here what we can do now in an automated fashion, within a few seconds, start to quantify that behavior in an unbiased, robust, and repeatable way. So this is Polyanotus digging its burrow. It's most interesting when you consider this compared to Maniculatus. Same exact setup. So what you're watching, that white thing is the mouse, sort of in cloud space. So you can see Maniculatus is just as active as Polyanotus. In fact, it's more active. It runs around more. This particular mouse is a bit funny because it's built itself this little ramp And sometimes you'll see it charging off the ramp and doing these backflips off the side. It is running around and running around. It's doing everything but burrowing. Okay? But it knows, well, it may or may not know, but the lights are going to come on in 8 hours. And all of a sudden, it builds its burrow very quickly. In a short amount of time, this guy tended to build 2 burrows, and then it sits in that burrow and doesn't do anything for the rest of the trial. Okay? So right away, you can already see some dramatic differences in the behavior, and we can use these sort of behavioral profiles from the 2 species and hundreds of individuals to start to quantify differences in the actual mouse's behavior that produces the differences in the burrows. So we can use these analyses to measure all sorts of things, like the rate of burrow extension. How fast do they dig when they dig? It turns out that Polynotus is indeed a little faster than Maniculatus, but the difference is only very small and only explains about 10% of the overall difference in burrow length. What you probably could have gleaned already from those videos is that the main difference between the two is the time spent digging. So Polynotus spends much more time digging than Maniculatus, and this result was of particular interest because it tells us that it's probably not something sort of mechanical, because they can dig at the same rate, more or less, but it's more having to do with something that I'll loosely refer to as a motivational difference. These Polynotus dig for much longer. The other thing you probably noticed from those videos is that when they start digging is very different. Polynotus almost immediately start digging when we put them in the boxes relative to when the lights come on. Maniculatus wait till an hour or so before the lights turn on and then quickly dig their burrows. So we refer to these as our eager beavers and these guys as our procrastinators. Okay? They both build burrows but at very different timing. But again, this is slightly consistent with the idea that there are differences in motivation. And this really got us thinking about maybe the big difference in these burrowing behaviors has to do with motivation to dig. So I just showed you this data. So we see a difference in the 2 in terms of the time they spent digging. Polynotus digs much more. They're more eager to dig when we put them in the boxes. Remember back to that data I showed you earlier, Polynotus digs more frequently in addition to building longer burrows compared to Maniculatus. When given the opportunity, they don't always burrow. And then the third piece of data, which I haven't showed you, but I'll touch on just briefly, is that there's a difference in burrowing timing, not just during the nighttime activity, but over development. So these mice, about 23 days of age, we can remove the pups from the mom because they can survive without the mom. It turns out if we take the mice and put them in a burrowing box, Polynotus, the first mice will start digging at 17 days of age. These are little pinky mice that can't survive without their mom, yet they'll build a burrow, and they'll build a burrow that has an entrance tunnel and an escape tunnel. It's just very small. Okay? Maniculatus, these little diggers that only have to dig a little burrow, the first ones don't start digging till about 10 days later. And it turns out that if you're now we can do this in our back cross mice, it turns out that if you're an early digger in development, you also dig longer burrows as an adult. So all of this data together suggests that there's motivation is a very loaded term, so I'll use it lightly with quotes, air quotes around it, that there's a difference in motivation in these 2 species. Okay, to end, I'm going to return to that gene I told you about earlier that got us so excited based just on its expression patterns that we see in the brains of these mice, and just tell you briefly what we know about this gene in mice, laboratory mice, and humans. So it turns out this gene that I've been calling Chrome5 or M5 is a muscarinic acetylcholine receptor, M5. It's the 5th member of this particular family and happens to be the least well studied, of course. But we do know some things about this gene. For example, in genome-wide association studies in humans, mutations in or near this gene have been associated with nicotine addiction. And if you knock out this gene in laboratory mice, it has a phenotype associated with morphine addiction. Okay? So this is a gene that's associated with addictive or motivated behaviors. We also know that it's expressed in dopaminergic neurons, so it's involved in the release of dopamine. We all know that dopamine is the feel-good neurotransmitter involved in reward. And it's expressed primarily in two main regions of the brain, or has been best studied in these two regions. The ventral tegmental area, which is well known for being involved in reward circuitry, and the substantia nigra, which is involved in motor control, spatial learning, and addiction. So this is what we know about its function in mice. We've gone as far to ask, is it expressed in the same regions in Peromyscus? Indeed, if we look at the VTA, this reward center of the brain, M5 is expressed in dopaminergic neurons. Here, this is a co-marker with dopamine synthesis, and they're co-expressed. So now what we're doing, which I don't have the results to tell you, now what we're doing is trying to use viral vectors to express this particular gene at differing amounts in particular brain regions in the little digging mice, and trying to ask, do they dig bigger burrows, and assaying those in those beautiful ant farm experiments we have now. So you'll have to stay tuned for those results, but I wanted to give you a sense of the approach we're taking, the types of genes we're finding, and trying to make these connections between genes and behaviors. And I would argue that if we're able to do this final experiment, make little diggers build long burrows, this is a result that would certainly tickle Darwin, both because it's a connection between genes, but importantly, a connection between genes and behavior, this idea that behaviors, just like morphologies, could evolve by natural selection if they're heritable.